Al, you want to do a partial zoom on this guy? Huh, looks like a hat. Oh.
Yep, that's
candy.
the sponge.
Toasted marshmallow.
Yep.
We want dessert. I think that's what we're thinking.
I'm going in a little bit farther.
Yeah, go ahead.
Okay. No, that's definitely an orange juicer. Put an orange
Does anybody know what a group of sponges is called?
Sponges?
Kitchen sink.
A herd?
A kitchen.
All right, go ahead and come wide out there, Al. Yeah, now we're on the edge of our seats. Can't just leave us cliffhanger like that.
Oh, we got a great chat in the science log on absorption of sponges.
Absorption.
15.
Oh.
Yeah, that's pretty good. Think we need to submit some of these new names.
Absorption.
So sorry, what was the group of sponges name called?
Well, we decided that it's an absorption of sponges. I don't think it's official.
It's rock here?
So until now, there was nothing official for sponges.
I think it's a rock.
Not sure. I haven't looked that one up yet, but some of the ones we've looked up so far tonight don't seem to have an official name.
Huh.
So, yep, that was a lone we just went over.
It's another orange juicer.
Yep. I think these are Hyolanema, which are the stock sponges, and their stock is very low on these guys. You can get a field of a muddy habitat like this, and it's like being in some kind of marine underwater crazy Alice in Wonderland tulip field. So many unbelievable.
Can't tell from that.
Ms. Clark is asking about collecting any of these sponges. What do you think, Meredith?
Let me check our log and yeah, if we could pause and maybe take a close look at this one that's up to the left, a little far forward.
Sure. Did you want just a quick look, or do you want a quick look in potential?
Let's do a quick look on potential sample.
Roger.
All right, bridge is holding.
All right. We'll sit down right here.
Orange juicers.
All right, video. And
Seeing
one moment.
A lot of burrows.
Yeah.
Ready when you are.
All right.
Going in. That juice has been overworked.
It's a great picture.
Yeah, I mean, see if Liz comments again. Because I'm not seeing a long stalk on this one, but I don't know if it's just because it hasn't grown a long stalk. What's our bio box availability?
Yeah, so in the starboard bio box, we have E and F, which are the larger ones, completely empty, and then we have D, which is one of the smaller ones that's also empty.
Okay, if we could grab just a piece of this and put it in one of them, that would be good.
Okay.
Okay. Sure. Or do you guys want to slurp? Would a piece of it would be kind of small, so it'd be kind of floaty. So, slurp.
Yeah. Well, do we have any slurps available?
Good question.
I'm
pushing it.
Right now, all of the slurps are currently filled. But if you wanted to put it in with another sponge sample if that's possible, or I don't
No,
know.
that's
not,
actually,
okay.
we
Sorry, I forgot
we
about
very
the
much have to keep
compatibility.
the sponges totally separate so
Okay.
that we don't have any cross of spicules.
All right. You can come full wide there, Al.
Sure.
Maybe I'll grab this and then we might have to pick up and move in case we swing.
See, I want to sample this because it looks like hyalinema, but that kind of flat orange juicy top does look almost like a carnus, which was a new species that was found a couple of years ago. And we will have to see when it comes up.
Yeah,
Okay. Okay.
that sounds great. Thank you. Sure. Yeah, that sounds good.
Want me to push
I mean,
it a
this
little
is
bit?
going to be a haircut on this guy. You want a part of it? Or do you want all of it?
If we can take just a part, that's always a nice do. But if it looks like it's just a size that may just come right up if we grab it.
Yeah, looks pretty.
Jess, you want me to push in a little bit?
It's all right. Yeah, thank you. I guess taking half of it would probably make it go through my fingers, so probably all of it will be getting you the actual sample. Is that okay?
Okay. Yep.
Brute force 5. Hopefully this doesn't.
One of our scientists at shore is chiming in that this would be another good candidate to pair with metabolomics. So that would be lovely.
Yeah. All right, we're just going to take it.
Squish.
Oh, orange juicer, you are stuck.
Oh.
All right. Well,
So we can actually let go of it now.
Yeah.
Because with it that stuck, it's going to be hyalinema.
So we don't want this?
No, I think it will like recover from its pinch, because that means it's got the really thick fiber optic stalk that is buried down in the sediment, and that is like a rope of fiber optic fibers.
All right. Well, okay. We gave it a new hairdo for today.
Yeah.
Do I have a second to zoom in and get a close look at that dew?
Yeah.
Probably want to pick up, but sure, go ahead, Al, if you want to.
Just let me see what happened.
Yeah. I guess you can kind of see that stalk down there.
Yeah.
Oh, wow.
That's the
tough.
fibers there. Yeah. And that guy will reinflate from his squish.
All right.
Okay.
Gonna come
cut
wide
out.
there, Al? Don't really want to show my hairdo. This guy. I mean, interesting that it's so stuck there.
Yeah, no, those
right, Aaron,
those
go ahead.
stalks are really stiff.
Science, just a quick ops thing. We are now 100 meters from the last transect, so if you'd like to do it, I guess it's the time. We're in our last hour and 10 minutes of the dive.
Yep, let's go ahead and do one last transect.
All right.
They're just on our bottom left. You can see too, their stalks are covered in crinoids, but you can see that they're, you can kind of see more of the stalk structure and how they form that kind of tulipy flower.
All right, we're gonna start with 064 100 meters.
Roger.
All right. Bridge nav. We have a 40-meter step bearing 064.
Roger. All right, I'm on 064.
So this would be Transect 12.
All right.
And have you started the transect officially, or just holler when you have?
How do you feel? Do you feel like we started officially?
Oh no, I was just getting in
I will-
position.
Okay, I'll reset the point
Okay.
then.
Yeah.
Oh,
Or we can keep going.
no, it's good. Let me.
We
Pretty
were
sandy.
moving so smoothly.
All right.
You ready?
Push the button.
For real? All right, here we go. Ready, go.
Yeah, I think I'm gonna increase that to 15. That's a lot of button push in there, hey?
I lost track again. Was it 12?
12, yes.
Thank you.
But Tom will get 2 more mud transects unless we come across some rocks.
I'm gonna give you a tug there pretty soon. I'll stand by here once
One of our scientists, Sure, was asking about whether the ones with long stalks or short stalks were different. They're all the same. They just don't have the stalk hasn't extended out on the short one.
Meredith, what's the species with the most morphology that just every time you, the most morphological differences that every time you see these 2 different types of the same species, your mind gets blown? You have to pick one.
Some of the shallow water Montastria corals come to mind. I would say some of the sponges really do that. Get some of the things that are like mycale, that they can grow in these really wobbly forms with lots of ridges, or they can grow as these kind of smooth dumpling shapes. And that's one of the reasons that they're such a nightmare to work with.
I thought you were going to say Swiftia.
Swiftia actually has the opposite problem in that you have, at this point, I think it's two genera and up to 3 possible families that have the same morphology.
Oh, is that what it is? Okay.
Yeah.
That's funny.
So it's that they all look the same, even though they're very different underneath. Whereas some of the sponges, you can look like completely different things, even though they are the same.
That's amazing. And how many species of rockfish are there that look so similar that it's almost impossible to tell apart unless you're a dedicated expert?
Oh, I think it's like 50 or something. It's there. And there's still questions about how many rock species there are. The lab I work in at Northwest Fisheries Science Center is still doing some projects where they're comparing species of rockfish, for which there's still an open question. So they've moved on from using mitochondrial markers for a long time, have been the like what we call the bar coding standard, and now they're using whole nuclear genomes. Jimmy says, "You mean so fascinating, not a nightmare?" And I said, well, fascinating and nightmare are often overlapping. Depends on how deeply in the data you're buried at the time. A deep sea soul.
Oh, bless my soul.
Mm-hmm.
And I've read that there's about a similar number of deep sea coral species that there are shallow coral species that have been identified. Is that still the case, or are we finding more deep sea coral species these days, and that's maybe a dated estimate?
I think that depends on who you ask. I know, at least last I had heard, Steve Cairns is of the opinion that there are more deep sea coral species than there are shallow. And just from a geographical standpoint, actually, that would make a lot of sense. Because deep sea corals and cold water corals just have so much more habitat available to them. And they grow from pole to pole. They've been found in every ocean basin. And so they probably are more diverse just because they have so much habitat available. Whereas tropical corals, even though they are the rainforests in the sea and you have huge amounts of cryptic diversity, only occur in these really specific habitats.
That makes sense. What do we know about the connectivity of shallow corals, the connectivity of deep corals? Is it kind of similar in their patterns, or what do we know about that?
It's very species specific. So it's in both groups you can have high levels of endemism, and what endemism means is how many species, the species that only occurs in one place. And so things like seamounts, both deep and shallow, can have high degrees of endemism where the animals you find there are only found there. But we do have other species that are found quite broadly distributed. There are some shallow coral species that you can find through whole ocean basins. And then we have deep sea coral species that can be found globally. And so it's somewhat species specific. When we look at populations, on the West Coast, we have some of these Swiftia twigs that we have studied. The species Swiftia simplex is one population coastwide. They put out so many offspring that there's the whole coast is connected up and down the coast. If we look at a group like Primnoa, you can have individual communities that are quite far separated because their larvae just don't move that far. So when we look at connectivity, we really have to say, okay, well, in which species? So it's a matter of picking the species you're most interested in.
Do we know about whether that has to do with larval dynamics? Some larvae may be heavier, some are lighter, some last longer in the water column.
It's a mix of what we call reproductive strategy, and that is how the animals reproduce. So in corals and actually most oceanic animals, you have two modes. You can have brooding, which is you reproduce and the little babies host kind of on the mom and then just pop off when they're ready and only go very locally. Or you can do what's called broadcast spawning, which is you broadcast all of your reproductive material into the water column. And at that point, it depends on how long the larvae stay up in the water column, how strong the current is, what season it is. So you do have some deep sea species that can go months in the water column and are carried quite far. You have others that stay quite locally, like deep sea canyons will often have very local larval retention. So it's one of these, the picture gets more complex every time you look at it, and but it is important. It's one of the things when we're looking at things like selecting habitats for protection, one of the things that you often want to think about is this a larval source for other communities? Or is this an area that new corals can land in and settle? And so those are all considerations for conservation.
Absolutely. And I don't know how much those kind of things are considered now. When I hear Fishery Management Council discussions, I don't think it's considered as much as I think it will be in the future. Since we're just learning about these things in the deep sea, it's such a new science. It's fascinating to think about how protected area decisions will change over the next however many years.
Yep. It's just one of those areas where we're building the plane as we fly it, and we're trying to make the most well-informed scientific decisions we can as we learn more about the life history of these animals. We've got little amphiptylum sea pens kind of starting to be mixed in here. There's a red fluffy question marks. We've had 2 go by so far that I've seen. And this is the Amphiptylum grandiflorum. That's the mud-dwelling species. And it's pretty cool. It's bioluminescent. So if you get it at the right time of day, in the right circumstances, and bump it, it can flash bright blue.
How do we know that a deep sea species is bioluminescent if we're always shining a light on it?
That would be a question for Gary Williams, who is the source of that particular piece of information. That one, I think they've had it bioluminescent when they've gotten it up on deck in things like trawls.
Makes sense.
Yeah, one of the shallow water cousins, the Tylosarcus, the orange sea pen, that you can see scuba diving up here in the Pacific Northwest, it is also bioluminescent. And so if you're recreationally diving and you see them, particularly they'll only do it at night. If you put your light on your chest and then carefully, gently run your finger up the stalk, they'll sparkle bright blue as well.
That's awesome. Are those the ones that the Tritonia, what are they, sea slugs, I think? Are those the ones that Tritonia eat?
Those are the ones that the Tritonia eat. And we actually have shallow here, another nudibranch species that feeds on them as well.
I love doing the experiment in undergrad where you can dye one of those sea pens all kinds of funny colors, and then the Tritonia eat them and they turn funny colors too. It's very entertaining.
Yep. One of our scientists ashore was asking if we'd seen any potato sponges yet. We have not. I think we're a little too deep. So sponges, because we don't know what a lot of these species are, we often come up with kind of fun descriptor names for them. So we've had, what is it, the shaggy dog sponge? And we've been discussing the moldy Swiss cheese sponge, but one sponge we found in the last couple of years we've been calling the potato, and it's a free living sponge. So it's not growing on rocks or anything, and it looks like a rock or a potato on the bottom.
Science. So as we kind of changed contours now, I'm going to change my heading. I'm not sure if you need this noted or not in your logs.
I'll make a note of it in my log that I'm going to pass off to Tom, but thank you.
Sure.
What's the new heading?
It'll be 045.
Thank you.
Getting into another steep section here all of a sudden. This one came up very suddenly, just like the other one, although this one is not quite as dramatic.
Yeah, I think we'll be creeping in along the wall of this canyon now. Right.
Mm-hmm.
Yeah, go ahead, Al. Okay. Sounds good, thanks, Al.
Thorny heads and tanner crowns and crinoids. when the group at Southwest Fisheries Science Center does their counts, sometimes if a species like a crinoid is so dense, they do cease to attempt to count all of them and just label them, I believe, as abundant. I think here we've got just enough that they'll just be counting lots. Oh, there's a nice sponge.
Oh, nobody. Nice. Thanks, Al.
I think some of those we call the IKEA lamps. It's really hard to hear. I'm back in my office with all the doors shut, but I can't tell how much of like my dog sound gets through. And I was like, quick, mute, mute.
An enjoyable amount of barking.
All right, I'm gonna do one more heading adjustment here. So I'm not making. Okay.
Sounds good.
Whoa.
See pen just throwing straight out of the wall there. Oh, wow.
More dramatic cliff.
Now
Okay.
the cliff got dramatic.
There's the wall.
So do you want the same of kind of facing upper crust and then continuing on?
Yep, that would be perfect.
Okay.
Yeah. I think we have.
Bridge now 10-meter step, same bearing. I'm trying to give you time to catch up, but I don't want it to lose momentum.
Sure. Yeah, that sounds good.
A hagfish. That's the first hagfish I've seen all day.
Pretty hard down at me. Yeah. Great, thank you.
Looks like we got another cliff edge here.
Yeah. It's gonna fly manual, so.
It's interesting how much like sticky mud is still stuck to this basically vertical surface. You can see clear spots where you can see the harder substrates, but there's still plenty of mud.
Okay. Lateraling off the surface now. Continue on the 4 or 5 and dropping down.
Bridge nav, 10-meter step, same bearing. Yep,
Those are
10.
some nice burrows.
Thank you.
There's a small rock there completely covered in those soulless sea cucumbers. Really interesting topography in here.
Sure is. Interesting looking thing up the top, purple looking.
Yeah, it's a
Oh, there's
deep
a
sea
puffball anemone.
Oh, okay.
It's pretty.
Yeah.
Nice puffball anemones. There's a little tube sponge just at the top of the screen, another deep sea sole, and a vertically hanging thorny head there.
I'm going to do a slight adjustment to 050. Roger. Roger. Bridge nav.
Great, thank you.
20-meter step bearing 050. Thank you.
Thank you.
Like there's probably a sea pen coming out of the mud in that cliff up there. But looks like it curls, so my guess would be Discoptilum.
Just about at the end of my leash here.
Come on, ship.
So for Jenny, who's asking where we are, I think we are still in the dogleg. We're approaching the bottom right-hand corner on the HYPACK screen, shows waypoint 1.13, if we want to reference it against the other map that Kurt made us.
Bridge nav, another step, same bearing.
How far away are we from the sanctuary right now? From Olympic Coast Sanctuary?
We are just outside. The head of this canyon is within the sanctuary, but we're down in the Biogenic 2 EFH area. That's just outside of the sanctuary boundary. And so because they've done surveys at the top of this canyon in the sanctuary, this was a great opportunity to get this kind of cross-boundary survey of the whole canyon and be able to work this deep.
That makes sense. And are you seeing many differences in the kind of topography and the geology and the biology just inside, just outside the sanctuary?
You know, what we'll have to do is kind of take out the confounding variable of depth. So we are seeing differences right now, but I think it's largely depth driven. But just having that depth driven
Aaron, you want to pull Argus a little bit
set
off
of data
just a bit too?
will
This
better
next
inform
move.
management because it looks at the canyon as a whole ecosystem.
All right. No, just maybe we'll bias the next one to the east a bit since we're kind of 15 meters off. Yeah. Roger.
As I wonder with these canyon walls, if one of the reasons that we aren't seeing like tons of coral on them, if they're a very kind of
Coming
friable,
up a
more
little bit
brittle
here.
rock. That's a really nice puffball anemone there. Looks like it
Is
might
that
have
the
a
official
crab
name?
behind it. I believe that is actually the official name.
That's an excellent name.
All right, wait.
Thorny head there. That's an umbellula growing out of the wall. Got lost from its compatriots above.
So I've heard that one of the great things about sanctuaries is some of them can act as sentinel sites, and I think that Olympic Coast is actually officially a sentinel site for a lot of things, including ocean acidification. So we can do a lot of monitoring in sanctuaries and really get a handle on how conditions are changing over time. But do we do any monitoring in the deep sea? Is that possible at all? Is it just too expensive to get to the same site over and over again? What's your take on that, Meredith?
There are some limited sites where they do long-term monitoring, but just due to the expense and how little we've been able to explore of the seafloor, a lot of the emphasis is still on overall exploration. Like we really just don't have a handle on what's down there. So there are some sites, particularly as they discover some particularly striking sites that do have more long-term monitoring. Monterey Canyon would be one, but we still have very much a focus on exploration because we just don't know what's here in a lot of the areas.
Please?
And Jenny Waddell, who is the sanctuary research coordinator, just reminded us that Ocean Networks Canada has a great monitoring setup with systems called Venus and Neptune, I think, that are anchored in the deep sea and constantly monitoring conditions there. National Science Foundation also has ocean observatories in some deep areas. And one of our researchers out of the Northwest Fisheries Science Center, Elizabeth Clark, said that Daisy Bank is a great place for deep sea monitoring. So there are a few places that we're starting to do this kind of work too. But absolutely, like Meredith said, that exploration has been a substantial priority. We have a map of deep sea coral locations that are known so far. Around the US and also internationally. And that map looks like there's a lot of blank space, and there probably is a lot of blank space out there, but a great deal of that blank space is what we don't yet know. So when you see all that blank space, you can't say, well, we don't have deep sea corals or we don't have these certain species out there because it just hasn't been explored yet. And there's so much that we just don't know about the deep sea that that we need to discover before we can put a lot of effort into monitoring in lots of different areas. But it's great that we have a number of places where we're starting to monitor and look at these long-term condition changes in the deep sea as well.
So that's end of Transect, everybody. Congratulations.
Yay!
Hooray.
Thank you.
We got 40 minutes to play.
Awesome. Well, let's continue to kind of slowly move along this feature.
Sure.
And there's a couple, there's a C-pen that I will try to give you enough advance warning that I'd like to take just a quick zoom in on and get a good look at. So like possibly some of these that are just coming above the lasers now.
Yeah, sure. If it's just a zoom, we don't have to stop the ship or anything like that. So
Yeah.
I can get us kind of steady and we can take a look.
And the last thing we wanted to note about monitoring versus exploration and combination in the deep sea
All right,
is
Al.
that uh,
Steady for you there. Just pop up.
just the Ocean Exploration Trust with the Nautilus and Hercules setup are really crucial to this work that we're doing. And they have a great partnership with Ocean Networks Canada too. So we just wanted to, again, say our appreciation for this partnership that we have.
Very happy with that.
Thanks. That's a great zoom. I think this is probably the deep sea species of Holothurians.
Yeah, go ahead a
This
little
is
bit more.
species we're doing some work in. But we're not actually collecting it from this depth zone because we do actually have plenty of specimens from these depths. So it's a really good view to see all those polyps out and open.
Can I get
This is a species that
chat, please?
All right, if you guys are happy with the zoom, I'm gonna keep moving since Argus is still swinging from the last move.
Yep, perfect. That sounds great.
Great. Yeah. Up we go. So my camera.
That species is one of the ones that can surprisingly, despite its size, suck down into the sediment and surprisingly quickly.
Bridge enough. $20 step 090. Thank you.
Meredith, did you say that that sea pen can suck down below the surface?
Yep.
I
Those
didn't
ones,
know
even
that.
though they can be a meter high, can bury themselves entirely.
Does it collapse or does it bury? Both?
So it uses its peduncle to basically drill a hole down into the sediment. And then when we have the zoom, if you look carefully or if you want to rewind on the video, you'll see all these little white dots all along the edge where the polyps are. And those are actually a specialized type of polyp that acts to fill the tissue of the sea pen with water or expel it all. So they will very quickly expel all their water and use that to pull themselves down into the sediment.
That's amazing.
These ones, when we've sampled them, we've been able to be quick enough for most of them, but we've had some other species of sea pens that can do the same thing, that you go to grab them with the ROV and just the simplest touch, and they're gone amazingly fast.
So Science, as we explore along this contour, is there any other kind of itemized objectives that you want to knock off your list before we have to leave the seafloor?
I'm going to keep my eyes open for any more corals and sponges that are on our list. That kind of sponge we passed while we were on Transect, if we saw more of those, we'd be interested in trying to collect one. And then I'd like to get a Niskin right before we leave the seafloor.
Okay, sounds great.
We seem to be in a pretty muddy zone, so I'm betting we're just going to explore for the last little bit.
And do you want to keep it at this contour, or you need any kind of upslope, downslope, preferably upslope motion?
We can contour a little upslope, kind of get a little shallower, see if it changes.
Sure. Great, thank you.
Meredith, do we know why crinoids and other species collect near these tiny ridges? Is there a change of current flow? Is there a fluid aggregation? Do you know what's going on?
In that case right there, it looks like there's a band of harder
those are
substrate,
10 meter
and my guess
contours,
is
right?
they can hang on a little better.
Yeah, I think that'll be good.
So
Probably
it's
like
just they
a
have a
north
better
move,
grip.
0 type. Roger.
Lots of critters burrowing around here.
Makes me happy.
Oh, there's a good thorny head there. Right under a C10. I'm always amazed at how many different colors the thorny heads can be, even when they're all the same same species. And that's actually some rockfish do that. That can be very dark red or very almost white, and they can change color depending on the light level they're in. It can make IDing them quite annoying.
Bridget, I have another step, same bearing.
Do we know if deep sea octopus change color the same way the shallow octopus do?
You know, I don't know. I know they can change their skin texture in a very similar way, but I don't know if they have the same chromatophore. Some of these thorny heads are quite small. When they first land on the sediment, they're actually usually a dark gray, and then as they grow, they morph to the red. And I think one of the last things to change is their fins.
I saw a few flatfish at the beginning of this watch, but I don't think I've seen many since. Rather than missing them.
Yep, I've seen a couple of the deep sea sole, but nothing else for a little while.
We're slowly going up slope.
We are,
Have
but
you
happy to
noticed?
change anytime you want to.
Yeah.
Oh no, I'm fine with it. I was wondering, Meredith, have you noticed changes in the community between 800 and 900 meters in that kind of range?
There hasn't been a too dramatic a community change with depth in here. What's largely been is driven by the substrate. So we've transitioned into this really soft mud, and so we've gone from seeing all of these different corals and sponges that were growing on the hard substrate walls, and now we're seeing all species that are soft substrate.
Okay.
If we go up a little higher, I think we'll start to see a faunal change, but not until we really get to like 600, which I don't think we'll get to today.
Right. It's curious because it looks like the oxygen minimum zone for this area is around 800 meters. So I'm wondering if once you get above that 800 meter depth, you start seeing different things because of oxygen levels. But
Bridge nav,
it's more
another step, same
substrate
bearing.
dependent. That makes sense.
Yeah, I think it's substrate and just also depth. In the deep ocean, we actually very commonly see species distribution changes with depth. It can be related to things like the oxygen minimum zone, but it's also just you know, different environments, different temperatures, pressures, and some deep sea currents can be quite stable. So you can have water layers that are you know, basically the same kind of type and similar water masses over long periods of time in some areas.
I've definitely learned a lot on this watch, and I love learning that the water in this area is some of the oldest deep sea water. That's a fascinating concept.
That's the they call it the global conveyor belt in oceanography, and they get the North Atlantic deep water formation that forms up around Greenland, where the cold, highly saline water sinks underneath this kind of sinks to the bottom, and then begins this journey where it travels all through all the other ocean basins, and finally it kind of comes back to the surface up here in the Pacific.
Yeah, it's just fascinating to think of water as old. I love that concept. Old and full of nutrients.
That they didn't really know how old until the 1950s, and quite possibly one of the only positive things to come out of nuclear testing was the ability to age water masses much more sensitively.
Oh, that's how it's done with nuclear testing.
Yep.
Also didn't know
They
that.
used deuterium, which is a form of hydrogen. And we started to learn just how old some of the water was. Oh, there we've got a swimming crinoid right in the middle.
Yeah, I've been
Just
waiting for one
settling
of
back
those.
down.
Did that just reposition itself to be in the middle of the shot, or is it just me?
Yes.
If only he was free floating for another hot second there.
Wanted to make sure got it turn on camera. Got some nice deep sea holothurians mixed in. These kind of almost transparent log-like objects on the bottom. They're deep sea cucumbers.
I've seen some.
In the 2017 expedition in Olympic Coast, they actually sampled some of those, and a taxonomist from Florida, who's a sea cucumber specialist, is taking a look at them right now, and hopefully we'll have some results in the next month or so as to what our clear species are.
Do you see those transparent sea cucumbers in lots of different areas, or are they fairly unique to the Pacific Northwest?
We do see them all the way down the coast, but they're a very deep species, so you only see them when you're working at these really deep depths. A few more right there.
How long have we got to go? All right. If you like.
Nice close up of the sea cucumber we were just talking about.
Oh, on the left, there's one of those fiber optic sponges for Abby. We had quite a patch of these sponges for a while. Another one of these kind of tulip gardens. And we might be getting into another one. These ones have been, a lot of these have been like this, where they look like they're sitting on the bottom, but they do have that really thick stalk right underneath. I just went over a little baby thorny head too. See, right now, what I'm sitting in my office with my face really close to the screen as I look for little objects in the mud and look for little critters. And it's one of the times that I'm missing the really big high definition screens in the van.
Yeah, I just have my laptop screen that I'm straining to see in. But there's plenty there to look at.
2 sponges.
It's been a great dive so far. Mm-hmm. Is that another orange juicer sponge?
Yes.
Yep, that's another fiber optic orange juicer. We have a lanternfish there swimming through.
Fiber optic orange juicer.
Sounds
How
very
long can I
high-tech.
make this animal name?
Yep, and Abby has just given us the species. It's Hyolonema populiferum.
That works too.
It will now also forever be known as the fiber optic orange juicer.
Do we have any sense for how old a sponge like that might be? I know sponges are complicated to age and lots of variables, but if you had to guess, is it more on the order of 5 years, 10 years, 100 years, 1,000 years?
You know, honestly, on those ones, I would not know where to begin to guess. So I have no idea. They're ones that I wonder, because they have those thick spicules that they use to attach to the sediment, if the material in that would be a good candidate for some type of isotopic aging.
Good question.
On the right, we just passed an Anthoptilum. And I think some of these white sticks that we see laying in the sediment here are actually the internal stipes of some of the sea pens we're seeing, sea pens that are no more. Some of these species, not the Umbellula, but these others, the Holothurians and the Funiculina, actually have this proteinaceous hard stipe in the center that helps support them. And I think that's what these white twigs we're seeing on the sediment intermittently are.
Do we know how old sea pens tend to get?
I think they are actually some of the faster growing ones. I know there is an interest, and there have been a couple studies to see on ones like the large Holothurians whether you can cross-section their stipe and use that to age them. But I don't know if that's actually been successful yet.
I heard Jenny earlier saying that there was a species, I can't remember which one we were looking at, that was probably about the same age as she is. And I love thinking about that. I love thinking about the over 4,000-year-old sponges, or sorry, corals that have been discovered in the Pacific Islands. I have an intern right now working in my office who's got a project going to compare the timelines of deep sea coral individuals, the ones that we know how old they are or can get, with human civilization. So what were humans doing way back when this 4,000-year-old coral settled in the Hanamokotea Marine National Monument? It's a really cool project to see what people were doing at different times as it was